Hello, everybody, and welcome to part two of Notable People in the History of Biotechnology. So, we ended with our two godfathers, our two fathers of recombinant DNA, Stanley Cohen and Herbert Boyer, and we're going to pick up in the 80s with one Dr. Ben Carson. So, Dr. Ben Carson grew up in the inner city of Detroit and Boston, and his parents divorced when Ben was 8 years old, and he and his brother were raised by their mom. Ben struggled in school a lot, and he was raised, and as a kid, he had failing grades, he had a low opinion of himself, and he also lashed out and had some behavior problems. But in high school, Ben started to turn himself around, and he eventually got a scholarship to Yale University. And he continued his studies at the University of Michigan Medical School, where he studied specifically neurosurgery, or brain surgery, as we would call it. After graduation, he moved to a position at Johns Hopkins University Hospital, where he later worked his way up to the director of pediatric surgery at the age of 32. Dr. Carson gained worldwide recognition in 1987 when he became the first surgeon to successfully separate conjoined twins who were joined at the back of the head. Now, when I say the back of the head, I'm talking about right here. They were joined right between the ear and the center of the neck. This is a very difficult surgery, and in the past, before this surgery, before Dr. Carson developed this surgical technique, which would then be used repeatedly, basically parents were asked to choose if those twins, those babies were conjoined, to choose which child they wanted saved if the surgery didn't go well. Dr. Carson saved both of those children, and in March of 2013, Dr. Carson retired as a surgeon. And he said he'd much rather quit when he's at the top of his game, and his retirement became official in July 1st of 2018. And basically what followed is he ran for president, didn't get elected, and now serves as the director of the housing of HUD under the Trump administration. Dr. Ben Carson, very successful individual, very inspiring story, and one of the most interesting individuals in our notable people, most notably because he's still around, alive and kicking, and basically having an impact on our world. Our next individual, this is Dr. Ananda Chakrabarty. I'm going to say that one more time. Ananda Chakrabarty. So, Ananda Mohan Chakrabarty, Doctor, PhD, is an Indian American microbiologist, scientist, and researcher who is most notable for his work in directed evolution, or genetic engineering, and his role in developing a genetically engineered organism using a plasmid transfer back to recombinant DNA technology while working at General Electric, and the patent that led to the landmark Supreme Court case of Diamond versus Chakrabarty. As a genetic engineer, Dr. Chakrabarty began working for General Electric, and he had developed a bacteria derived from a Pseudomonas genus, and now known as Pseudomonas putida, capable of breaking down, basically eating, crude oil, in which he proposed to be used in cleaning up oil spills. General Electric filed a patent for the application for the bacterium in the United States. It's listing Chakrabarty as its inventor. But the application got rejected by a patent examiner because under patent law at the time, living things were generally understood to not be patentable subject matter under Section 101 of Title 35 U.S. copyright law. And this was because living things were thought to be natural and they couldn't be patented. And so a lawsuit happened. General Electric and Ananda Chakrabarty sued. And so the Patent Board of Appeals and Interferences agreed with the original decision to basically say, nope, not going to work. We can't patent a natural thing. And appeals overturned the case, but the appeals court turned the case over into GE and Chakrabarty's favor, writing that the fact that microorganisms are alive is without legal significance for the purpose of patent law. Sidney A. Diamond, the Commissioner of Patents and Trademark, then appealed to the Supreme Court because they said, no, the law says this. And so the Supreme Court was argued on March 17th of 1980 and decided on June 16th of 1980. The patent was granted to General Electric and Dr. Ananda Chakrabarty on March 31st, 1981. Guys, this is a long story, but it is a big deal because Ananda Chakrabarty and General Electric won this case in the Supreme Court. It meant that other companies would be able to patent their genetically engineered bacteria and living things. That means that they would be able to profit off of their genetic engineering work. That's a huge deal. This opened the door to all types of companies coming in and being a part of the biotechnology revolution. Which leads us to Dr. Robert Hirsch. Another company that was trying to get in on the biotech revolution was a company called Monsanto. And at Monsanto, Robert Hirsch started studying genetic engineering and model plants like petunias because they were easy to grow and hopefully easily to genetically engineer. It stood as a good model because it was easy to handle, it cultured, it was well studied genetically because of the economic market for colored flowers. What Hirsch learned while studying the petunia made it possible to be successful in genetically engineering soybeans a few years later. In January of 1983, Robert Hirsch was ready to announce the expression of a specific genetic trait that was added to petunia cells. This trait was resistant to a specific antibiotic in transformed petunia cells, which demonstrated gene transfer and gene function in recombinant DNA in the lab in live petunia cells. This was a big announcement. It meant that recombinant DNA and genetic engineering could be performed on plants. In the past, they thought it would only work on simple organisms like bacteria, but Robert Hirsch proved that we could actually genetically engineer plants. So after leading Monsanto's plant tissue culture and transformation efforts, Hirsch became responsible for a small shareholder agricultural development partnerships and public-private technology cooperation programs. Hirsch saw that Monsanto's technologies developed for the U.S. agriculture were valuable and relevant to developing countries because developing countries, farmers have almost the same problems as U.S. problems. So his idea was to then take the technologies that he helped develop and spread them worldwide to help pass them out and teach others how to use these genetically engineered soybeans, corn, and cotton. Incredible. Dr. Norman Borlaug. Dr. Norman Borlaug is a personal hero of mine. He was born in Iowa in 1914. He grew up on his father's farm, and he attended 8 years of school in the same schoolhouse. It was a one-room school. And he earned a bachelor's of science in forestry, a master's degree in plant pathology, which is the study of plant diseases, and a PhD in plant pathology and genetics from the University of Minnesota. For 30 years, Dr. Borlaug worked as an agricultural researcher. His efforts focused on improving wheat. Later on, his efforts focused on improving corn. Later on, his efforts went to fighting climate change. Dr. Borlaug won a Nobel Peace Prize in 1970 for his recognition of his work for improving the living conditions of millions of people in developing countries. He's known as the father of the Green Revolution and continued his battle against starvation in Africa until his passing on September 12, 2009. And our next notable person, Dr. Ian Wilmot. Dr. Ian Wilmot was born in England in 1945. He earned his bachelor's degree in agricultural science and his PhD from the University of Cambridge in 1971. His early research at Cambridge University led to the birth of the first calf from a frozen embryo who they called Frosty. His frozen embryo research led to his work at the Rosalind Institute in Scotland. Since 1974, Dr. Ian Wilmot worked at the Rosalind Institute in Edinburgh, Scotland, and he gained recognition worldwide when his team of scientists successfully cloned an adult mammal for the first time. That clone was named Dolly the sheep and was born in 1996. Dr. Ian Wilmot continues to do research on cloning and also is hoping to figure out how to clone human embryos for stem cell treatments of degenerative disorders such as diabetes and Parkinson's disease. And that ends our list, everybody. So what we're going to be doing over the next couple of days is you guys are going to be asked to identify these individuals in a flippity, in a Quizlet, and on a quiz. Please refer to all the materials that I post into Google Classroom, and I will see you much later down the road.